Maybe we'll do what that group just had before us and have an exam right now. What do you think? Yeah, I like that idea. What's wrong, Portia? Okay, so today I'm going to take you through some beginning of hormonal regulation, and I'll talk a little bit about ubiquitination. Then when we have a break, I'll give you some general feedback on your mid-semester exam, plus also the current general feedback on your report part A. And just note that your report part B topics are already up on Views. You'll select those in the lab this week. Okay. That general feedback that I give in the lecture today, I'll be doing that on each campus, and then that'll go up on Views probably Thursday afternoon as a hard copy. You can have a look at it. I'd strongly recommend you look at it because I give general feedback to everybody, not just those who need it the most, because even those who think they've worked hard cover everything and miss little things. And I'll cover that today as well. We're dealing with, as I said, hormonal regulation. So I want you to think about this part of the semester, which is only 3 lecture topics to go through, essentially, for us, and there's plenty more we should be dealing with. There's hormonal regulation, so outside regulation of cellular metabolism. Now, we spent the first half of the semester looking at inside the cell, getting stuff in there, doing things with it, looking at a general balance. And, to be perfectly blunt, we actually didn't do a very in-depth job. I'm just giving you the language so you understand the key concepts, because it's pretty complicated, isn't it? That's why you did that report part A. I kept giving hints about report part A, looking at EC number, enzyme commission number, looking at metabolic pathways using KEGG. Report part B deals with metabolic manipulators, drugs, compounds that affect cells like hormones do. You might find an EC number, but more than likely you will not, because these drugs or these compounds are classes of molecules, and I'm going to hint at those over the next few weeks. Classes like different types of G protein drugs, signal transduction drugs, lipid-based drugs, for example. They're all different classes, like the enzymes were. We're dealing with what I call extracellular messengers, because not only are they hormones, they could be cytokines. Now, cytokines are essentially immune cell messengers. They're usually shorter distance, shorter acting, but they affect you just as significantly, if not more. Think about when you have a virus infection of some kind. You feel quite lethargic, don't you? What your body is doing is moving energy away from your normal activity, whatever that is, to defence, because you have a general amount of energy available, and during an attack, that defence is making antibodies, making immune cells, other molecules. That's what we have to deal with for metabolism. They need the energy to do that work. Once you've dealt with that attack, mitigated it in some way, then the energy comes back to you. And just think about the time frame that happens, especially this time of year. And most of you get flus, they get all those special considerations. You were sick for how many days? If you had a vaccination, what do you find? Maybe not as sick for as long, because you've already pre-primed, everything's ready to go. And I'm going to interweave that as we go to today as well. These extracellular messengers as hormones or cytokines, there's a few other odd ones. We talked about nitric oxide already. That's an extracellular messenger. Most of them are peptides. Most of them are steroids. Not necessarily the same as your metabolic manipulator. That could be a drug, a synthetic drug, or an isolated drug compound. Much of your functioning starts up here. It's integrated information from the external environment. We talk about an advanced cell. How you take that external environment, either as photons in your eye or taste in your mouth, sound is in your ear, feelings, convert that to an action potential down the nerve cell to where? To here, to integrate it, to decide what to do. Now, when I was teasing you a minute ago that, oh yes, we're going to have an exam, your heart rate probably went up a little flutter. Your brain perceived there's a stress, but that's a perceived stress, not a real physical stress. Your perception of a metabolism exam right now. What happens? Oh, okay, it's not real. All right, so then it abates. Think about that when you turn over the exam booklet in the final exam. Now, you've seen your final exam timetable? Yeah. Is that right? Is it Tuesday that it's on? Okay, so now we have to work to that date. In your brain, you integrate that information and you tell the rest of your body what to do. But not only is there external environment signals, there's internal environment signals. What your fat cells are telling you to do, what your gut cells are telling you to do, and other integrations that go on inside. All of this, you have to think about your cells in your brain have to make a decision, which is the prominent activity. And then it's going to send out a signal to say, this is what's going on. But as it does, I need you to think about it's not just one signal. Your cells are getting signals all the time, which is the predominant one. I'm going to use the analogy I use a lot. You're at home, you're playing on your phone or on your computer, someone's in the kitchen saying dinner's ready. What do? Keep playing on your phone or your computer. I know, because I have one of those too. Until the signal from the kitchen gets loud enough, it dominates what you're doing to say, get in the kitchen or you get no dinner. So what you've done is you've made a decision initially not to respond to that signal until the signal gets loud enough. Your cells are integrating tens of thousands of signals all the time. It's deciding which one to respond to. And that's what we have to think about as we go through now. It's not just one signal, as with some of these simple schematics are showing, it's multiple all the time, which is the dominant signal. We're seeing that go out to other actions, like we're seeing in the range in your brain. It's around micrometers, through the synaptic cleft, from your brain, through your hypothalamus, down to your pituitary, anterior, posterior, then secreting hormones. Some of them get into early functional activity, like your thyroid. Others go down to your adrenal, affect your stress response. And some of those go through the blood vasculature. Now that means they're already going to have to deal with a difficult environment. Insulin's one of those that has to be disulfide bonded to make it stable, to last long enough. And in your report part B, you have to talk about the half-life of that molecule. Drug analysis includes half-life because you want to know how long it lasts, because the longer it lasts, the more efficacious it may be. And maybe too long, it becomes traumatic for the patient. We're seeing here hormonal signal coming into this pancreas. We're going to talk about islet cells, alpha, beta and gamma islet cells that secrete things like insulin, glucagon, somatostatin, regulating appetite, regulating blood glucose. We're going to see how insulin's secreted now. All of those impact what you're doing, but they're just simple hormones. Hellishly complicated, but simple. But this is the scale range. Micrometers down to centimetres and metres. What, from my brain down to my adrenal gland is almost a metre, isn't it? Down here at the base. So I've got to send a signal through the blood vasculature. The hormone that does that work, ACTH, has a very short half-life, broken down very quickly. Your brain sends a neuronal signal down to your adrenal, we'll see today. It releases adrenaline, the catecholamines. That has an incredibly short half-life, seconds and minutes at the most. But if it lasted longer, it could be dramatic for you, couldn't it? Heart palpitations and you'd die probably. So you don't want these compounds to live too long, some of these messengers. Others you want longer. We're comfortable with the beginning of this concept of extracellular signaling. I'll move to the next one. Okay. Here is our friend insulin. Here is our pancreas, sitting up here nicely. What's it doing? It is manufacturing in these little islets, like islands, hence the word islet, islands of cells specifically designed. Here, the beta cells producing insulin, the alpha producing glucagon. There's the two counter-regulators. This thing, somatostatin, regulates appetite. So the delta cells regulate appetite, functionally controlling and controlled by your gut. Insulin is a peptide hormone. It's around 5.8 kilodaltons. It comes out in this sequence. I need you to look over to the right for a start. It comes out as a long polypeptide with an amino signal sequence. I showed you that before, didn't I? If it's the beginning, it often gets into the ER, maybe packaged up in vesicles, then the cis-medial trans-Golgi, and then we see in advanced cell how it pushes off to the membrane with snare-snap proteins. But along its path, it has to be post-translationally modified. The first thing it does, as we saw earlier, is the signal sequence is cut off. I'm in the ER now, I'm inside a vesicle, I'm not part of a membrane. So this is the insulin, not the insulin receptor I'm going to show you shortly. While I'm in there going through the cis-medial and trans-Golgi, I need to develop these disulfide bonds, 3 of them, to help stabilise the molecule because it's going to go outside the cell. When we get close to secreting outside the cell, we're going to clip off this region called the C-peptide. Now, many assays measure the C-peptide and not actually insulin. It's a surrogate measure of what's in your bloodstream. That depends on the assay. What happens then is this mature insulin now, with our B and A subunits, here it is here, the A and B chain, are packaged up in a vesicle inside your islet cells. Which ones? The beta cells, and they wait for a signal to release insulin to your bloodstream. When does it release insulin? When your blood glucose goes up. But we're going to see the signalling events to take that up, to actually release the insulin component. It's a fairly straightforward molecule. I mentioned the disulfides, but it requires calcium. There's an interesting fact yet again. Calcium to activate enzymes to do some work. What's calcium doing? Activates the proteases to cleave off the C-peptide. What's a protease? Like we saw in your gut, it cuts peptide bonds specifically. In this case, it cuts out the C-peptide in your beta cells. If you get a blood glucose rise, then insulin gets secreted out. That's next semester, all the SNAP, as I mentioned. And then you start to see the insulin run around your bloodstream doing what? Interacting with its receptor on the cell surface. That could be muscle cells, fat cells, liver cells, whatever the case is, it needs to interact with this receptor. Now here's a key point for you. These beta cells producing insulin. The question is, first of all, how much? Is it mature? Is it appropriate? There's my insulin in my bloodstream. It has a half-life. How long does it last? Then my insulin's got to run around the bloodstream and find a receptor. Here's insulin. Here's its receptor. How many receptors on the surface of the cell? That depends on the time of day, what you're doing. Are those receptors functional? That's why diabetes is so damn complex, because it's not just making the insulin, it's getting the signal into the cell. And I've got a slide to show you to give you an idea how complex it is, and a nice little animation to show you what really goes on, not just some still versions like this. So you're trying to alter other cells. What does insulin do to those cells? If it gets the signal through the receptor, what does it do to those cells? What does it tell them to do? Yeah, it puts on more glucose transporters to, say, suck up glucose, particularly the liver, but everybody else, doesn't it? Whatever's in your bloodstream as glucose, suck it up and put it into storage. Your brain's using it all the time, isn't it? We're kind of comfortable with this one. So we've just made insulin. We put it into a vesicle. It's ready in your beta cells, waiting for you to have a meal. Okay, let's have a meal, shall we? Shall we go to the next one? Is that yes? Sorry? I'm not shouting. I'll turn the mic down if you're worried about me shouting. Is that better for you now? Is that better for you now, Charlie? Cool. All right, can we go to the next one? I want you to look at the slide first, don't write. This thing on the left here is a beta cell. Don't write, look at the slide. I'm watching. There's a beta cell. There's its nucleus. What does this particular cell do? Here it is. It's just made insulin like we saw a moment ago and stuck it into vesicles. There they are sitting around near the plasma membrane. Lipid bilayer, lots of insulin. Later on we'll see SNAPs, push them up, squeeze up the membrane, push out insulin. But how do you get to push that insulin out? How do you signal that event? I've made lots of it, hopefully. Well, first of all, you've just had a meal, haven't you? There goes the lollies, little afternoon snack, ready for metabolism. What happens to your blood glucose? It goes up. Now you have to tell your beta cells your blood glucose is up. Okay, my beta cell has a glucose transporter already on the surface. There it is there, the GLUT2. When glucose comes in, what does it do? It starts glycolysis in your beta cell. Now, be careful, don't write, just listen for a start, I'll repeat this. It starts glycolysis. First, it traps the glucose, hexokinase we've seen before. There's a glucose 6-phosphate. It runs through glycolysis, the citric acid cycle, oxidative phosphorylation. What's it done in the beta cell? Generated lots of ATP. Okay, I've generated lots of ATP. What is a beta cell supposed to do? Well, it doesn't contract, doesn't do anything else. All that ATP, you see the square bracket, arrow up, I've done that all semester. Increased concentration of ATP in the beta cell stimulates, look at the board, stimulates this transporter. You see this purple thing? That's an integral membrane protein. That's a potassium transporter. Listen, watch, don't write. What's that potassium transport doing? As soon as you have elevated ATP, closes off. Why would it do that? It's busy pumping potassium out, but all this extra ATP closes it off. Because you've closed it off, you've altered the charge differential. I introduced that last week, didn't I, on our inner membrane of our mitochondria. I've got less positive charge on the outside. I've got now a differential in charge in the membrane of this beta cell. Okay, I've done that. It charges that, it depolarizes. What does it do to this character down here? That is a calcium pump. It's voltage sensitive. So as soon as the voltage changes, what does it do? It opens up. And what does calcium do? It's a cofactor. It stimulates somebody. It comes barreling into the cell, increasing concentration, arrow up. It tells my little vesicles that are near the membrane, snare snap, squeeze them up, push out insulin. So let me repeat that. To get secretion of insulin, first you have to take insulin into the beta cell, alter the ATP, increase it, close off the potassium channel, change the voltage, open the calcium channel, put calcium in there, now release insulin. That's just after you had a meal. And have you stimulated anybody to produce glucose transporters yet? No. You've just released insulin after that meal. How much glucose? I'll come back to you in a second, Charlie. How much glucose? Depends on what you've been exposed to, how much sugar's in your blood, and whether you've got issues with your beta cells. Yes, mate? As you write, when you have a meal, your blood glucose goes up. Before anything else, your beta cell takes up the glucose, runs that glucose after it's trapped it through glycolysis, through the citric acid cycle, through the electron transport chain, makes lots of ATP. Too much ATP in a beta cell closes the potassium channel, which is busy pumping potassium charges out, that positive charge. Because you've closed that off, you've changed the voltage differential on that plasma membrane. That change in voltage, which incidentally is the same effect when it gets near your synapse. So when you've got now an axon coming down to a dendrite, it's a sodium potential almost to the end, and just at the end it changes to a calcium channel. The same kind of one you see here. When it changes to that calcium in the synaptic cleft, it causes the fusion of the membranes with acetylcholine, release of those. The same phenomenon you see here. So lots of ATP, shut the potassium channel, change the voltage, because no longer you're pumping potassium out regularly. Because the voltage changes, you open the calcium channel, calcium floods in, what does it cause? Snare snap proteins and membrane fused, insulin into your bloodstream. Is everyone clear on that? Was that slow enough for you? No?
No, it is. I think you want to move on. No way.
No. Now, there are drugs. Here's a hint for you already. There are drugs that affect these pathways. One I have down here for you, sulfonylurea drugs, close potassium channels. Which drug you pick. It may not be an actual drug, it might be an enzyme kind, but most of them are drugs on your list. They're drugs or pharmaceuticals or drugs derived from natural products. And this one's used to treat type 2 diabetes. Now, I'm not asking you to do an assignment on diabetes because it's done to death. It's too much information. You'll be flooded with information. I'm going to crystallise down the real critical stuff in diabetes, given I had a number of students go and work in the Joslin Diabetes Centres in Harvard. Name dropping, just a little bit. So we've altered these channels. There's our friend calcium. Didn't we make a point about calcium all semester? A cofactor to stimulate cytoplasmic activity. Alter somebody. Yes, you need ATP to do the work, but calcium's the cofactor. You have to ask the question for your report, part B, how does your compound, your metabolic manipulator act? What does it do to cells? Are we ready to move on? Not yet. Now just note the terms before I do move on, because I'm going to be dropping these in and out as we go. Down here, you note here, it's used to treat type 2 diabetes. There's a number of types of diabetes, type 1, type 2, has different names in the old days, early onset, juvenile. There's also pregnancy-induced diabetes. You have to think about how diabetes actually functions. What's the input to it? You'll notice here it says overcomes insulin resistance. Here's a drug that tells a beta cell, if you're not doing your job, responding to blood glucose, going through these pathways, I'm going to do it for you and release insulin for you. Is the receptor affected yet? No, we haven't looked at that yet. It's the release of insulin that's a problem in type 2, not the response of insulin at the cells. That kind of makes sense? Okay, can I move on now? Good. And this is where we have to worry about receptors. I'm going to give you a little kind of Cook's tour of receptors generally. I want you to look at this ugly schematic here. That yellow and blue tram track you see up there is the cytoplasmic membrane of a cell. I've got a student looking at that at the moment in hyperspectral analysis. That is very fluid, very mobile, lots of proteins in there causing all sorts of drama for us. In there happens to be receptors, often proteins, polypeptides. They might go in and out of the membrane a number of times. They might be aggregates, they might be dimers. Lots of examples of those. There's a hormone, might be a cytokine, usually a peptide. Usually a peptide or peptide-derived hormone. Adrenaline is a hormone. What's that derived from? An amino acid. It has a receptor. That receptor is embedded in the membrane. That receptor might change shape as a function of the hormone binding to it. A drug you select might actually bind in the same place and stop the receptor activating. It might stop signal transduction we're going to talk about, which is this pathway down here. Things like G protein, cyclic AMP, IP3. Protein kinases are in there as well. Here we are seeing cyclic AMP. They are termed second messengers. They will activate the cytoplasm of the cell, usually through calcium, not always. They'll tell the cell to get busy with whatever machinery they've got, whatever enzymes. If you keep hitting the cell, you'll often see a signal get all the way to the nucleus, and I'll give you that example shortly, where you start stimulating gene expression to put more machinery out there. It's like, I'm getting the signal to make cars, I need more cars, what do I need? More workers, more factory. So what do? Build more factories, get more workers. But then if I don't get a signal, what do I do to my factories? Start closing them down. It's the same kind of economy. These other characters are things like these ion pumps or ion channels. We just saw potassium and calcium. Some pumping calcium out, some sodium back in, whatever the case is. Sometimes hormones stick to those, like acetylcholine in your synaptic cleft, or the one that controls muscle contraction. Then we have these guys, hormones. I want you to write down there mostly steroid hormones. They're hydrophobic hormones. Cortisol, estrogen, testosterone, classic steroid hormones. They are hydrophobic. They generally, in your system, do not have cytoplasmic membrane receptors. They slip through the lipid bilayer, they get into the cytoplasm. Here's my steroid. It's gone into my cell now. Inside my cell cytoplasm, it hooks up to a protein like a flick knife. That flick knife opens up. That complex then goes to the nucleus and alters gene expression. That's a transcriptional regulator. Write it down. Steroids do not have in your system's extracellular or cell surface receptors. The steroid has to get into the cell first before it finds its receptor in the cytoplasm. And those steroid receptors often get all the way through to the nucleus immediately. They don't generally alter the cytoplasm. Once they get into the nucleus with that flick knife, you see the green one I mentioned there, they alter transcription. They alter gene expression. They might increase or decrease. If it's testosterone, they're increasing things like muscle accretion, protein deposition. They might change your behaviour if you're male or female. Some of these others are things like thyroid. I'm going to talk a lot about those in the next few weeks. Coming down here, the thyroid, a bit of a H-shaped gland. That's a metabolic regulator. It up-regulates or down-regulates your metabolism based on what you do, how cold you are, how warm you are, those things you're going through. So this gives you an idea of the very simple models of receptors on the cell surface. Now, in the case of this hormone binding to the cell surface receptor, you don't necessarily need the hormone receptor complex. So let's pick a different hormone. Let's get here it is. What's that, my coffee card? That's my hormone. Let's say that's adrenaline. Very short half-life, breaks down very quickly. What does it do? Sticks to its receptor on the surface. Goes in and out of a membrane about 7 times, 11 angstroms in the first domain, causes a change in conformation. Now this thing signals an event. Doesn't mean the hormone, adrenaline, has to go into the cell to get a response. Some do, some don't. Adrenaline doesn't need to. So in this case, as I have down here, the ligand, I want you to underline that word, the ligand, which in this case could be the hormone, the ligand could be your drug. Anything that sticks to a receptor, please write this down, anything that sticks to a receptor is called a ligand. Could be labelled with isotope, could be labelled with fluorescence markers, could be labelled with anything, but if it sticks to a receptor, it's a ligand. And there's different types of ligands. They could be chemical agents, pharmaceutical agents. They can alter things like differentiation, metabolism. All of those events can go on. And before I get to the next slide, I want you to notice very simplistically, this is a very, very, very simple diagram. It's showing you one cell surface receptor, showing you one ion transporter and one steroid hormone. All of your cells have hundreds. And they're not just sitting there waiting for a signal. They come and go, they come and go, doing all sorts of things to you all the time. And that's overlaying how much hormone you're dumping out into your system. Does that kind of make sense? Can I move to the next one? Okay. And this is what I'm segueing into. It's not the thing with the 2 wheels you run around and fall over a lot. This ugly brown thing, there's the purple nucleus down there as a cell. You see that blue thing up there? That's the receptor. Those little red dots are the hormone. Now, this is a cell surface receptor. Here we have a cell with not many receptors on the surface. Think for a moment. The glucose transporters, what do? We stuck them in a membrane. There they are. They're stuck in the membrane, and then we put them to the cell surface, didn't we? We brought them back in again when we didn't want to take up much glucose. These are the same kinds of phenomena. If you want more receptors on the surface, the cell puts them up there. If you want less on the surface, you take them down again. They're still there inside the cell, but they're not getting the signal from outside the cell. So what you're seeing here is this context where I might not have many receptors, but I have lots of hormones. I have lots of insulin dumping out, but not very many receptors. So what does your cell do? Oh, think about it. It's like you out in your room playing on your phone, and there's a noise from the kitchen, dinner's ready, and it's not loud enough yet, is it? I'm too busy on my phone doing my own thing. But here we are now. What's happening is your acuteness of hearing is increased because you're hungry. Playing on your phone all evening, and now a voice from the kitchen, dinner's ready, phone down, off you go, get something to eat. Because you're more responsive to that signal. And that's what you're seeing in this concept. You're seeing a stronger response because your cells are seeing more hormone. The number of receptors on the surface has increased, which can also do the opposite and decrease. What we see down below is this down regulation. From the kitchen, think about that for a moment. From the kitchen, I've got something to eat. I go in there, there's a nice feast, stuffing my face, and now I'm full. Do you want to eat some more? Come on, have seconds. Come on, a bit more. No, just one more bit. You ever watch Monty Python? Just a wafer before the guy explodes? Oh, come on, you should watch Monty Python. It's fun to watch. He explodes. He had one last wafer. So you're full now. Do you want to eat any more? No. Down-regulated. Are you listening to eat anymore? No, I want to go back and play on my phone. That's what I'm doing. I'm down-regulating the signal to eat food and go and do something else. Your cells do this all the time. Overwhelming signal, they down-regulate, which is what you're seeing here. This overwhelming signal, they take the receptors off the surface and put them back in the cell, like we saw with the glucose transporters. Not enough glucose in the blood, we have to then take them out, so we're not sucking up glucose. Excuse me. This alters, and I want to underline, the target cell sensitivity. This is going to be type 1 diabetes, where we alter the receptor responsiveness, not type 2 where we can't get enough insulin dumped out. And we see this occurring for a long-term exposure. You know, if you're in the kitchen all the time, oh, food, I just couldn't stand anymore. I've got to go away and just veg out, ruminate for a while. I've had enough. Can't eat anymore. That's the signal you're seeing here. And this occurs over longer periods. So the more hormone, the more signal you get from the kitchen, usually, unless your stomach's taken over, usually you don't want to eat as much because there's just too much food on offer. We see that with hormones. I want you to underline pharmacological agents. That's assignment part B. What do those drugs do to hormone receptors? Do they up and down regulate over time? What's the dose and time course? They're critical things in biochemistry and pharmacology, the dose and time course. How much binding goes on? That's the bit where you want to jot down how sticky is the ligand or hormone for the receptor. If it's really sticky, it'll stick there and do its job. If it's kind of weak, half-assed, then it's not going to do much. And what you also want to underline is this about different cells do different things. Because here is our receptor. Let's go back one slide. There is a receptor. That might have a different isoform. Write it down on different tissue. So you might get the right hormone, you might get a receptor, but it's not exactly the same receptor, so it's not exactly the same response. That's tissue-specific receptors. You see that particularly during stem cell differentiation. Oops, wrong one. I want you to underline this other word too, affinity. I'm not going to take you into too much receptor biochemistry. It gets a little bit difficult in the time frame we have and where you're at in terms of what we're dealing with. But affinity is how sticky it is, how specific it is. If you've got a key to this room, not a swipe key, if you've got a key to this room, that key should fit nicely, shouldn't it? If your key doesn't fit too well, it's actually hard to unlock. So I hate using that lock and key model because receptors change conformation. Some ligands do too. So it kind of fits in, it feels kind of cozy, that's good. Then I get a response. If it doesn't fit in too well, it might come in and actually sit there but do nothing. I want you to write the term down, an antagonist ligand. Antagonist, like I am. Versus an agonist ligand, which gets in and stimulates the receptor to respond. And I want you to write down another term, partial agonist, partial antagonist. Your compound, part B, might be one of those. It gets in there and doesn't do a very good job, but enough to slow things down, so to speak. We comfortable with these concepts? Is that a yes? Can I move to the next one? All right. I want you to start down here at the bottom. What you have along the x-axis is the relative concentration of a hormone, a drug, molecule, whatever it is. It's your ligand, increasing concentration. Write that down. Increasing concentration, doesn't matter what values we're dealing with today. On the y-axis, what have we got? The fraction of maximal binding or cellular response. That could be anything. How much NADH, which you have to measure today, or this week I should say, in your cells. How much NADH, cyclic AMP, could be how much calcium, how much potassium, could be anything that you want to measure. Whatever it is, you're measuring something that changes over time. In this case, over concentration. What you see down here is as you increase concentration, we're not seeing a linear response. You're seeing this physiological response where it kind of maxes out around this. That's just a nominal figure, it could be anything. But I want you to notice the shape. See how it doesn't take much concentration of our ligand, boom, and it pretty much maxes out. Why is that? Let's go back here. I've hit my receptor. Let's go back here. I've hit the second messengers. That's it. They're told to work. Bless you. I've gone to work. There's the factory. Get to work. Can they work any faster? No, that's as fast as they can go. To go faster and make more cars, I need more machinery, more workers. That's why I go back to transcription and translation. So I've maxed out. That's the physiological response. Now let's see what happens if I take the receptors out and look at them in a tube. I want to know, for example, a drug, for example, that might bind to this receptor, what happens to it? And the real problem is not so much the shape of the curve as how do you get a number that's meaningful? Well, you need to do this bit down here. You see where these dashed lines are? A bit like you do in your standard curve. I run along from the OD value and I drop down to the concentration, yeah? Well, what's what they're doing here? They're coming across, dropping down. What I want you to write down, that's the 50% value. It's like how we measure titer, which I won't go into today. I might go into it later on. How many antibodies you have? Well, I don't exactly know the maximal range, but I can say this is where it's maxed out. I'm going to look at the 50% value. That 50% value tells me here it's about 1 concentration, whatever that is, of my ligand to get an effect. So now I can have a meaningful number, because won't that depend on how many receptors, what type of cell, the signal transduction, all the events in a normal responding cell. And what happens in a cell is that other schematic you see off to the right, which is the amplification. One of a few little animations for today. This amplification of receptor signalling. The gong means we're still going, I hope. Let's put this thing over here. Now, I just want you to notice, before I show the animation, this thing here, adrenaline. Okay, adrenaline sticking to its receptor. Here it is. A very powerful, potent ligand. Very short half-life, gets into your bloodstream from your adrenal medulla, bam, hits its receptor. Now that's a 7 transmembrane receptor, in and out of the membrane 7 times. This thing gets into the first subunit, 11 angstroms, poof, everything happens. Respiration rate, heart rate, blood glucose, everything goes crazy. What does it do? Well, at 10 to the minus 10 molar, one piece of adrenaline molecule, one molecule itself, hitting its receptor can amplify 3 adenyl cyclase. Remember that molecule that's stuck in the membrane? This happens to be a G protein coupler. GS-alpha zooms along the membrane, activates AC, write it down up there, not DC, AC, and not the band either. What does AC do? It amplifies more molecules like cyclic AMP, lots of them. Get down here, what do? Each of those activates protein kinase. In this case, protein kinase A. What's that do? Phosphorylate somebody, busy as. What do those do? Each one of those activates another enzyme. Each one of those activates another enzyme and product. So then you've got this amplified signal, particularly from adrenaline, very, very fast. Does that make sense? So now your cell can respond quickly, and that's all of your cells, which I might turn the lights out for this little bit. And if you think about the moment that I said to you that we're going to have an exam in here, and that doesn't help much, but we'll go on with it. How quickly your perception of stress changed your heart rate and your respiration rate. How rapidly that went from your brain to your adrenal to stimulating adrenaline release to stimulating functional activity in cells. This is the lipid bilayer. This is outside the cell, here's inside the cell. Here's a receptor, this character here. It's actually a polypeptide going through the membrane. Nice little hydrophobic domains looping around. Here comes a hormone, happens to be a growth factor. Now, growth factors generally work as dimers. That's why you're seeing blue and red. When these characters get together, they actually form a dimer of receptor. Now, adrenaline receptor doesn't need a dimer. It just needs its receptor, its hormone, bam, things happen. But growth factors, some drugs like report part B, might need a dimer. And when they have a dimer, they might need a partner for their particular receptor. They have formed a dimer receptor. I've aggregated. Now think about that kitchen sound again. Sound from the kitchen, dinner's ready. Still on your phone. There's another sound from the kitchen, someone else saying you better get in here because if you don't, we're going to give you dinner to the dog. So that's two signals amplifying. Does that kind of make sense? Coming together. What have you done? You've aggregated this signal inside your cell. Those two guys happen to be kinase receptors. What do? They phosphorylate each other. Oh, that feels good. Don't do that. So I'm going to phosphorylate you, you phosphorylate me. What do I start? A signal cascade. What do? We amplify the signal, just like we saw with adrenaline. We start telling the cell to get busy with whatever it's doing based on that signal. We've started ligand stimulation. And let's go back on. We can either work through one receptor, one ligand, or we can work through a dimer. What are you doing? Activating the signals. What I haven't talked much about is that stickiness, how affinity. That's for next semester. Things like KD, dissociation constant. Your drug, your pharmaceutical agent for report part B might have a binding affinity. That could be critical. How sticky it is for its receptor. That drug, and I need to alert you yet again, I will a few times, because it might stick to a cell surface receptor like we're seeing back here, might be a particular class. Unlikely to have an EC number unless it's an enzyme, but a class of drug, like a G protein signaling drug, for example. What kind of class is it? You must have that in your report part B. Is that clear enough? What kind of molecule is it? Is it hydrophobic, hydrophilic, is it peptide? How does it work? Its mode of action. So now we can get this response. We've seen amplification of signal. We've got an idea of how things work at a very basic level, because as you can imagine, it gets more complicated inside the cell. Can I move to the next one? Is that a yes? Okay. This is closer to what we are in the real world. This should be an exam question. Don't write too much on this one. This is for your information, because the textbook and me, what we're doing is crystallising down the core concepts. This is a lot of work, and what we're seeing up here, here's insulin. I'm going to show you the receptor in a little video in a moment. There's insulin sticking to its receptor. This is a signal transduction cascade. Write it down. A signal transduction cascade that we know of. I got this last year. They give me updates every year when there's new molecules involved. What does this tell me? When insulin gets stimulated, it's altering all of these molecules, things like protein kinase 3, FIN, APS, CD1, RAC, PD kinase, CAP. All of these guys interact. What's going on? Well, it's like being in a factory. Foreman's running around, foreperson, someone else is running around. They're all doing stuff, interacting. What are you going to do? Make a car, hopefully. Hopefully you're coordinated. If you're not, what do you make? Bits of metal. You have to coordinate that event, which is critical in signal transduction, because if any of these characters, which are mostly polypeptides, are not made correctly, don't do their function, don't respond to calcium, what happens? You don't make your car. In your case, you might be diabetic. Type 1, for example. Alteration in any of these functional elements. What we see down here are things like mTOR, elongation factor, all those things that start regulating gene expression. There's some of my favourites, C, Jun and Foss. I worked on those years ago. Proto-oncogenes that involved in early activation, the first few minutes of activating a gene, usually less than 10 minutes, involved in stem cell differentiation. What do? Alter gene expression. What have you got here? The glucose transporter. You can see there's interaction between glucose uptake through the transporter and the insulin receptor, which is kind of critical for us because you need to link those two together. The bit I want you to write down is down the bottom, which is kind of critical. Now, this is recent research. There are other things going on, like hypoxia. What's that mean? Oxygen, because now your cells don't make enough ATP. Ubiquitination, I'm going to show you in a minute, put up above that, protein turnover, protein half-life, like you should have got for report part A. The thing we talk about in advanced cell, hopefully they do in molecular biology, is microRNAs. You want to write down there epigenetics. MicroRNAs regulating activity of polypeptides, particularly in glucose metabolism. Now I've got a web link up here which doesn't want to work in this system, so we're going to go the other way. Let's go here. I'd encourage you to watch these at some point. This one's a bit noisy.
Australia is facing an increasing epidemic of type 2 diabetes. There are now approximately 1 million people living with diabetes and around 100,000 new diagnoses each year. Obesity is thought to be the primary cause of type 2 diabetes in people who are genetically predisposed. In part 1, we explained how inflammation in type 2 diabetes destroys the pancreas tissue and dramatically decreases insulin production.
Don't panic, part 1, I'll show you next week. Okay? We're going to do it in reverse. We're dealing with the receptor first today.
Here we describe the insulin receptor in type 2 diabetes.
What we have discovered is the molecular detail of how the hormone insulin binds to its so-called receptor protein on the surface of a cell and instructs that cell to take up glucose from the blood and to store it as an energy source. The receptor itself actually spans
No gloves,
the
no
cell
glasses.
membrane, so it has a part outside, a part inside, and insulin binds to the part that is outside. Insulin is a small molecule whose structure was determined a long time ago in 1969, but it's been unknown how it interacts with the receptor, and that is what we have discovered. And its interaction I've characterized as a molecular handshake as the two proteins dock together. When insulin binds to the outside parts of the receptor, it brings the two halves of the receptor together, and at the same time, the two parts inside of the cell unwind and make contact. These two proteins
Creaky receptors,
then
eh?
activate each other.
This activation allows the insulin signal to enter the cell. Specialised proteins interact with the activated receptor just below the cell surface. The receptor activates these proteins, and they in turn are able to activate multiple other proteins in a long signal cascade. This complex signalling process eventually tells the cell to take up glucose for use as an energy source. The long signal cascade allows tight control of the signalling process, as it can be turned on or off at many different points along the way. In type 2 diabetes, we believe insulin binds to its receptor normally, but the signal is not sent into the cell and the signal cascade does not develop. This is known as insulin resistance. This means that glucose cannot enter the cell and blood glucose remains high. Over time, high glucose levels damage many different organs.
Insulin, as you know, has been used to treat diabetes for a very long time. But all of those formulations and types of insulin that have been used in the diabetes context have been designed and formulated without any knowledge of the way in which insulin actually interacts with the receptor. And now for the first time we've got a picture of that, and we can use that into the design of new insulins that could be used for the treatment of diabetes. So we could imagine, for example, the design of insulins that act for a longer period of time, design of insulins that act more quickly, design of insulins that are more stable upon storage, and that's where we like to go with our discovery.
The key thing of that is understanding the notion that it's a molecular handshake. It's protein-protein interaction. It's not lock and key. They're not fixed in time and space. And there's all these events, as we can see down here, that might alter the functioning of that signal, which is where things get more complicated, which is basically like your brain interacting the signal from the kitchen and wanting to play on your phone, where you integrate it, decide which is the predominant activity or pathway you're going to take. Can I move to the next one? Okay. The thing I have to talk a little bit about is the protein half-life. This is because report part A, we didn't spend enough time on the half-life of the polypeptide, how it was made. I showed you videos of how it was made. You should have done this in functional proteins and genes. Now, this is a function of what kind of amino acids that are in there. If you have things like methionine, glycine, alanine, serine, threonine, valine, you tend to increase the half-life of that polypeptide more. Disulfide bonds increase the half-life of that molecule more. If your drug, part B molecule, is a polypeptide, they might be altering it to increase its half-life, or maybe the opposite. Destabilising things like isoleucine, glutamate, taurine, proline, phenolate, all the ring-structured molecules, arginine. You can get down with lots of arginine just to a couple of minutes of the half-life of that molecule. The other things that change are what we call the end rule. A protein's half-life correlates to what's the amino acids on the N-terminal end of that amino acid, particularly if it's open and available. Things like methionine, serine, alanine, these guys alter the half-life significantly. These guys on this N-terminal reduce it significantly, phenylalanine, leucine, aspirin, lysine, arginine again. So you can see those molecules, their position in the polypeptide, how many of them alter their half-life. That's what chemists generally do. Glycosylation increases. What's glycosylation? What kind of groups? Carbohydrates, good job. Glucose molecules, for example, different kinds of sugar molecules alter the half-life, but they give it a unique structure and signal. Please note that. More glucose molecules, more hydrogen bonding, more unique signals. Talk about this a bit in advanced cell. What it means then is you learned how to write the letters when you were in kindergarten, didn't you? A, B, C, D, E. Now you have a signature. Well, a polypeptide is the letters. Glucose molecules is the signature. It makes it unique for you at that point in time. And your signature's changed over time, hasn't it? Your first one when you were in primary school looked pretty average, didn't it? Now it probably looks like mine, just a scrawl. That's what sugar moieties do to them. These are all important. Things like acetylation alter the half-life of the molecule as well. How long it stays there within the cell. There's the acetylation process. That happens a lot for molecules to alter what they do. Hemoglobin lasts about 110 days. How long does a red blood cell last? The same time, basically, 110 to 150 days. So that gives the half-life of the cell, not just the polypeptide. Can I move to the next one? Did you cover this in FP&G? A little bit? Good, nice reminder. The other bit is ubiquitination, little animation. Now this is important because this has a role for half-life inside the cell, not just of your proteins, I'll do the talking, not just your proteins or polypeptides, but also peptides that you introduce to the cell. Now this is also, I want you to jot down, also important for your immune cells, particularly B cells and macrophages, taking in bits of virus, hacking them up and presenting them on the MHC or HLA. What do? Well, you have to make a polypeptide first, fold it like a Sara Lee cake, put it together, disulfide bonds if it's going out. You might have to fold it inside a very unique environment. So you put it into a barrel, this thing called a chaperone molecule. Big polypeptide, in that barrel you can change the environment, change the pH, osmolarity, all sorts of things. In there you can fold, that often requires ATP. We'd use this molecule in advanced cell actin fibres. They're the same ones that cause that contraction with myosin. We can see them beautifully in images, can't we, Portia? There's a cell migrating. Now that particular actin molecule has a half-life. It's going to be broken down. It has an N-terminal amino acid that says, now it's your time. You're going to be recycled. What are you going to do? You're going to get ubiquitinated on that end. That's this molecule here, ubiquitin. It latches onto the end and it kind of latches on like a scrum. They're all lined up like the Congo. That polypeptide now is targeted for breakdown. It goes into this barrel called a proteasome, a special barrel where you have proteases that cleave this molecule into fragments. I want you to jot down usually 8 to 24 amino acids long. Now that's significant for your immune system because when your immune system sees any bacteria, virus, anybody attacking you that's based on proteins, your immune system fragments them into 8 to 24 amino acids. That's the sequence that you present on the surface of the cell. The cell says, this is not me. You need to start attacking it, making antibodies to it, for example, which is going to come back and bite you after we have a break. Because after we have a break, I'm going to talk about ELISAs, how you use antibodies to detect molecules. And often we use this system in model systems to make antibodies to those peptides, which are 8 to 24 amino acids. What did you do in that proteasome? You fragmented this thing, ready for complete recycling or write down for immune cell recognition of foreign polypeptides. You're also going to write down not just foreign polypeptides, but self. Now that's kind of critical because when you start developing from utero to about age 4, your immune system's starting to recognise what's you, not to attack you. To not attack you, it has to sense what is you. So that later on, when it senses there's something coming in that's not you, it will attack that. Does that kind of make sense? And that's why the early stages of your life, and this is to those who have children, gonna have children, those first 4 years, it's kind of critical to challenge your immune system without overwhelming it, so that your immune system gets to recognise what's you and what's not you. Now, I used to carry on about daycare when my kid was going through daycare. It was horrific. She used to bring home every disease known to man. It's bad for us as parents, but good for her, because she was challenged all the time. She's less likely to develop autoimmune diseases later on, like the Japanese tend to do, because they're all worried about sterility, sterile foods, keeping everything sterile, particularly during the early stages of development. That's a major problem, because when they should be busy learning about self, that is your immune self, they're not. Later on in life, that immune cells, well, I'm getting bored, sick of playing on my phone, I might go and attack something, like my own tissue, like my joints, like my pancreas. Because I'm not busy defending you, I don't have the memory for that, I'm going to go after something else. So keep in mind, this functional activity is not just for metabolism, it's what's used in other systems. Are we done with this one? Yes, mate. Sorry, what?
Directed proteolysis. What's the word?
Directed proteolysis. So what you're doing is taking this protein, this green thing here, and you're saying you are now targeted for destruction. So you're not targeting every molecule, you're only targeting those that have all these ubiquitin molecules stuck to them. Everybody else is okay until ubiquitin's on them, then bam, your time is up. Does that make sense? Okay. All right, done with this one. Yes? Time for a break. We started a bit late because of our predecessors being a bit busy with an exam. At 10 past, I'm going to start giving you feedback on your mid-semester and your report part A.
I'm Naja.
Hang on a sec. Can you hear me up the back? Yes? All right. Uh-oh's the word. Let's start with the mid-semester exam. Up on views, there's a notice to say that the mid-semester exam is worth 10%, right? And as I said on views, or at least I've set up a system to calculate your mark out of 10, that is 10%. Now, you did a will assay. For that will assay, unfortunately, views doesn't give us much opportunity to fiddle with the type and dynamics of the question. So the will assay had to be out of 3 or whatever. So you ignore that because every question was worth the same value, that is 0.5. Now, mid-semester exam, open book, take home, collude, coerce, whatever you want, that's up to you, but you're testing yourself. And some said last week, oh, it was a bit challenging. Yeah, it might be, because it was taking you outside just what we're dealing with, getting you to do other things. How many multiple choice questions do you have, including the will assay? 20. That's bugger all, isn't it? Hardly any, given that the normal for multiple choice questions is about 90 seconds, if not less if you're doing medicine. That gives you ample time. Indeed, when I look at how long it took the average student to do it, it was just over an hour, which is about what I calculated. Some people took longer. That's totally fine. It means you're looking around doing things. Some of you barreled through it because you might have found things on the web or got it from somebody else. That might be your problem in the final exam, because all of those questions are designed to take you beyond, to cement your knowledge and understand the language. Even the will assay questions, asking about alanine, PEP, whatever, are around what those issues are. Now, the good thing, as I said multiple times, is the final exam is not based on calculations. There's none in there. How many multiple choice in the final exam? 60. They're based on lecture material, what I'm covering in the lab. All through semester, after each topic, I put guiding questions up on views. Some of you are looking at that, having queries and questions, that's fine, but we get to our review lecture and maybe a tutorial, that's up to you if we decide to have one, in semester break, in StuVac I should say, because your exam is Tuesday afternoon? Morning. So there's not a lot of time left, is there? We have to start planning our attack for that exam. What I was pleased with was last year the average is around 5.5 for the mid-semester exam, and this year it's 6.9, which could be a good thing or a bad thing, depending on your perspective, which I thought was useful because hopefully it means you're talking to each other, learning with each other. That's a good thing. If you're not checking yourself and making sure that you know the work, then you're the only one cheating yourself, because it's open book. I asked you to talk to each other, to use different resources. So that's going to be up to you. We'll find out in the final exam, won't we? Those questions were fairly straightforward, I would have thought. A couple of hiccups with IT. We sorted most of those out, bar 1 or 2. Now let's get to your report part A. This is the interim feedback from the examiners. They're still going through, I think there's another 30 or 40 papers to go through. Some of the things, and you probably know some of the things you didn't do when you look at the rubric. I'll summarise a few for you. Majority of students did not include figures outlining the biochemical pathways. Didn't we ask for a figure or a schematic? Didn't I keep harping on that? Didn't I say go to KEGG or any other website because one figure is 1,000 words, that'll save you time. You can refer to it. Does it affect a particular metabolic pathway? You should have spoken to that. The other thing, you have Word or some equivalent on computers. Spelling and grammar is automatically checked. You have this grammatical check in the tutorial systems the university pays enormous amounts of money for. Didn't look like much of that was used. That's going to hurt you because there's a fair amount of marks if you look at the rubric for just spelling and grammar, isn't there? That's about being proficient and being sensitive to detail. I mentioned in the preamble to the report part A, take a pen or a pencil under each word as you read it. We are all guilty of skim reading, everybody, But if you don't do that, some of the text makes no sense because you brought it together, it makes no sense at all. The aim of these reports are to distill down information. That's why it's 1,000 words. Distill down information. The key points, not just here it is, slap it in, that's okay. It doesn't make sense. You have to read, and I have to say the key word is comprehend. Understand what you're reading. If it's not enough to read just that passage, read around the topic. That's what you do at university. Those are critical elements. What other things? Well, you know, things like half-life, cofactors, all the things we asked for in the overview. Some people didn't use the headings that we asked for. That's going to hurt. What else? Referencing. You know, most of you that I know of has done scientific literacy, haven't you? Which I think should be renamed scientific illiteracy. It's gone backwards, not forwards. I've mentioned many times in those tutorials, which some of you attended, many did not, about citing statements with a Vancouver format immediately after you cite that particular statement. Many, many statements were cited without any references at all. It's not your work, is it? You're summarising someone else's. Unless you're at the bench doing the work, you have to cite someone else. That's what you need to do. So you're going to get hammered for that. We asked for Vancouver referencing. We went through that in the lecture. We went through that in tutorial. That didn't appear. Some other format, which we don't know, appeared. Some people kind of thought 1,000 words means you can write 1,000 words on each particular heading. It doesn't mean that. You have to distill the information down. This is metabolism. It asks for you for biochemical pathways. I know that's not pretty. It's hard work. It's complicated, but that's what you've got to do. Does that make sense? Sometimes you have to do jobs you don't want to do, but you've got to do it. We didn't do that, some of you. Other things. You know, we're getting you exposed to the real world, the research sector, the diagnostic sector, the treatment and the policy sectors, to get a feel for what those languages are, how they use biochemistry, to give you exposure, because that's where you might want to work, for example. Don't want to be in a white lab coat. I don't want to work in a treatment hospital of some kind. I want to develop a drug, or I want to regulate some process, politically or otherwise. That gives you exposure. But you need to write to each topic area. And if we say clinical signs and symptoms, it just means, well, someone had a red rash. I mentioned in the lecture and tutorial, that's not enough. The differential diagnosis, what we're going to talk about a bit later on with ELISAs as well, and for your module 4, that's what you're up to. Analysing the protein in the urine sample, there's presentation information. What does that tell you? What the patient's saying to the doctor as they walk in. Well, you need to work out what's going on. With the protein, look at the sample, look at the presentation information. It's not as simple, oh yeah, there's my beverage, it's already got a protein concentration, 0 or some other value. That's easy, isn't it? Now you've got an unknown. You have to diagnose someone that might die if you don't do it correctly. Differential diagnosis is critical. The other things that come up all the time, I said you have to go to e-journals, the library, your library, you're paying fees for to get access to all of those journal articles. Wikipedia is free. It's a good place to start, but it's not where you should finish. Wikipedia is not a reference source. You have to go to the primary reference. You're going to get hammered for that. And too many web links instead of e-journals, not going to help you. And we said current. Well, some people didn't look at current. My external examiners looked for each of those topics. They know them pretty well. They look up. If someone says, I can only find the last 10 years, look up. Oh no, there's one just a few weeks ago or a few months ago. There are current. You may say not. We beg to differ. Depends on where you look, not just on Google. We asked you to look at policies inside and outside Australia because metabolism is global. It's not just based in Australia. Look globally. Jobs are global for this kind of activity. The other things you might want to worry about, for example, are the aspect, the biochemical process you focused on for your particular disorder. What does it do to the patient? What redundant pathways come in? What's going on in those individuals? And in the prognosis, we asked you to talk about what their life expectancy is. How many people are afflicted? the percentage of the population. We need statistical information to make decisions. Didn't see much of that in some places. You will need to have that for report part B. In report part A, we asked for an EC number, an enzyme commission number for the class of enzyme I kept going on about. The class. That class of enzyme is not just one enzyme, it might be a few that do exactly the same job. That's a class of enzyme. For report part B, it's a class of molecule, unlikely to have an EC number. Does it affect certain metabolic pathways? A signal transduction molecule as a class. A lipophilic molecule, that's a class. You need that for report part B. What else? You know, I just saw a confirmation of candidature for a master's student this morning come in. I have to mark a lot of these things. First thing I see is an image or a diagram, a schematic. What's that for? If you pop an image in straight away without talking about it, if we gave you a textbook with all images and no text, you go, what does it mean? Text first, then figure. Does that make sense? Describe, discuss, briefly introduce the figure. Don't just give us the figure and say, here it is, you work it out. No, that's your job, to tell us what it means, to crystallise it. When you use a figure, if it has little coloured things like green crosses or red triangles, you need to tell us what those are, what's going on in the legend of that component. Those are critical as well. You must have a legend for it, not just there's a figure I found from where, the web page. What does it mean? Pull the figure out and the legend, does it describe what's going on without the body of the text? It should. But with the body of your text, it enhances it, doesn't it? And you should introduce it first. Those are the things you should learn in scientific literacy. Because if you don't have those, you won't get a job. You won't be clear how you write. It'll just jump around. It makes no sense, like that document I read this morning. So I've got back to that student, rewrite it. You've got till midnight tonight. You have report part B. I'm giving you general feedback. Hopefully by Friday I'll have all the responses in. I'll be checking turns in, late submissions, and I hope to release the results on view by Friday. I thought Monday, but I keep forgetting Monday's a holiday, so Tuesday at the latest. The other key bits. Up until this morning, I was going to say nobody changed the dates on their particular journal articles. Examiner called me up. Somebody changed the date on their journal article. Why the hell would you do that? It makes no sense. If you're prepared to do that, it means you're prepared to bullshit somebody about a job or something else, isn't it? They wouldn't trust you. Don't change dates. It is what it is. Science is what it is at that point in time. Don't fudge things. That's going to be to your detriment long term. They will catch you out like we do. Because I said we'd check, and we do. What else? In the overview, you should be contextualising, crystallising, distilling information. That's important. Because if you don't do that, it's just this big, long, gobbledygook garbage that makes no sense. That's your job to do. You should think of that first sentence as a literary hook. If you read a novel, the first sentence either grabs you or it doesn't it? If it doesn't grab you, I'm not interested. Same deal. Contextualise what you're talking about. What's the issue with this disease or disorder? Same for part B. What's the issue with this drug or compound? What does it do? Not here it is. What's it do biologically? How does it help people or otherwise? Those are critical. Now, in some instances, a lot of people don't pay attention to the things we say. So we say, see you next year. You guys all right? Cool. What else? We asked you to link back to metabolism, the compounds we're talking about. Link back. How does that disorder affect fats, fat metabolism, carbohydrate metabolism, amino acids? You need to link it back, not just here it is. That's why a pathway, a schematic will help you, particularly. You know, some of the figures and tables were kind of less relevant. You have to ask yourself, like I mentioned to a student this morning, zooming about his presentation for his COC, be ruthless. Does that diagram or schematic, that figure, really help me tell a story, or is it just a pretty picture in space fill? That doesn't impress me, neither examiners, and I'll be looking at a number of reports as well. What else? Acronyms, symbols, colours. You should try and articulate what those are. Here is an acronym. BSA, what does it mean? Did you articulate what BSA stands for? What's BSA stand for? Bovine serum albumin. But if you're in a different field of activity, BSA might stand for a motorbike, might stand for a sore bottom. It's different for different fields. You must articulate that. That's the skill you should be learning at university. What else? Too many books. I said, don't give me a lecture or the external examiners a lecture on biochemistry. Let's get down to the grunt stuff. It's essentially a case study. What else? Reviews, I said you should start to look at. It helps you summarise the data, but you still should go to the primary source. How many should you have? Well, we kind of used a rule of thumb, didn't we? How many was a rule of thumb? A dozen or so. 40 is a bit too much, isn't it? Are they critical? Well, they might be, but you need to identify. Half a dozen or less? That might be a bit of a weak report, mightn't it? What else? In diagnosis, for example, I talked about differential diagnosis, weaving your way through, trying to work out what the disease is. How do they do it? Well, you might want a pathway to do that. That's useful. I asked for statistics particularly. How much of the population is afflicted, but also the prognosis for the patient. If the patient's treated or untreated, what happens to them? Is their lifespan short? Do they have to make sure they don't expose to sun, for example, with the porphyrias particularly? Those things have an impact on the patient, which is for you as well, because as metabolism graduates, you need to recognise if you screw up metabolism in some way, the patient's going to change, aren't they? What do they have to deal with because you've done something to them, as in drug or otherwise? Oh, and we go on. Now, before I do go on, please note, this is not for everyone. This is general feedback. And even students who thought they did a really good job, there might be something in here to just enhance it a bit further. That's why I give this kind of feedback. I mentioned things like treatment, things like adverse events in the tutorial. Yes, this drug goes in the patient because it's the only thing that keeps them alive, but the drug causes nausea, diarrhoea, for example. That's an adverse event. You need to talk about those things, particularly in metabolism. Any new treatments? I've mentioned things like gene therapy. I mentioned last week things like mitochondrial therapy, where you take healthy mitochondria from one patient, give it to the cell, the nucleus of the offspring, to give them healthy mitochondria rather than damaged mitochondria. The other thing in policy, we talked about things like treatment applications. How do you treat? Well, is it an injection like you saw a minute ago? Is it a nasal spray? Is it oral? How do you treat that patient? It's simply changing their diet or combination or exercise. That's a critical thing in metabolism as we go through this part of semester particularly. Some of the big issues overall, which I've already talked about on and off, when you write these papers, be consistent. It's easy for us to tell when you've cut and paste. Not only does Turnitin do that, and I'll be checking those the next few days, but the font changes, the style changes, and those external assignment help mobs that you pay money for, we know exactly when they help you because they can't write the way we ask you to write. We know when you're getting that help. And we know that's going to be a problem for you long term. That's why the universities around the world are saying don't do it, because long term that's going to be a serious problem for you. Because HR, when they look at your CV and all your introductory letter, know immediately. We train for that stuff in CSIRO. We know immediately when someone else has written it and you haven't written it. But you still should correct the grammar. You should write in your style, not my style. I'm a minimalist, as you probably will know. And lastly, to finish off, the word count. How many did we say? 1,000 words. You should work to that. Did that include the references, the legend, headings? No. Did it include the cover sheet? No. That's just the body of the text. You should aim for that number of words. They tell me. Views tells me everything. It tells me when you're on there, tells me when you watch videos, how long you watch these recordings, which places you're on for long periods of time, the time that's on there. All of your mid-semester exam, it told me how long you spent on each question, how long you took overall, how many times you responded to that question. It tells me everything, which I don't really want to know, but it tells me. So when someone says, oh, this didn't happen, well, I can check. No, yes, it did. All of those things it tells me. So be careful when you do things like your mid-semester exam and don't follow instructions where it says, use your patient number. If I use some other numbers, will they notice? Afraid so. But that also means I know who you are. And it means that process, your activity, is highlighted electronically, which means forever and a day, we know that you're not going to follow instructions or maybe not do what you're told. Does that make sense? The system is like that now. You better get used to it. If you don't follow instructions, they'll track you down and they'll know you're the one. That's how it works. So what's good to copy and paste is bad long term for knowing who you are. This will go up on Views 1 o'clock Thursday. I encourage you to read it again, check yourself, check the rubric, check everything, because report part B. Yes, mate. Let me ask you, why do you think it was hard? Indeed. And what do we say about policies? What kind of things? Not just symptoms, but the nature of the disease, when the patients need to be treated or diagnosed, and if there's an impact on things like the PBS, the Pharmaceutical Benefit Scheme. If I'm going to give somebody an injection, is there a policy on that? If I'm going to use a nasal spray, there's a policy on that. All of those things are policies. Now that's difficult, I believe, for you guys because you've never done that kind of work. Would that be right? Never looked at policies to try and understand the language of them. Would that be a fair assumption? Yeah, that's why this report's for, to get you outside to see what the real world looks like. Not just a textbook on metabolism, not just my lecture notes, but how the real world functions. And that's why you're doing report part A. Report part A to have a go, make mistakes, learn from them, take the feedback and take that into report part B and do a better job. Does that make sense? That's why I split it up into two, not here's just one. Mid-semester is straightforward because you have plenty of time. You know whether you got it right or wrong. There's just multiple choice. The answer's in front of you. The report part A and B is split up to give you feedback. Remember part B, we don't have a lot of time. That's end of semester submission and bam, we have to go for it. Mark like crazy before the final exam. So please take note of this input, what you need to do. For policy, we want to look at what's regulating. For the drugs you're looking at, most of them are drugs, not all. What's the policy on them? Well, it'll be about dose, time course, who's going to get that drug, when should they get that drug, who's going to be able to use it, that is, apply it to the individual. Is it oral, injection, some other way, patch? Those are all policies. Does that make sense? And the reason I'm doing that to you is so that when you graduate, you understand how complex our system is, that when you develop a new compound, you're doing your research, you need to have an eye on the bigger game, because not only might you get employment there, for example, if you invent this new molecule, and yes, it works really well, but it can't be used because you can't get it to the patient. That was a problem with gene therapy when I started on that, getting it to the patient. Well, that was a problem we've been solving over years, and it takes 20 years to get to that point. And you need to look to the future so you keep your jobs, pay off your mortgages, do all the things you want to do, not just have it for a short contract and that's over. Does that kind of answer your question in a roundabout way? Okay, I'm done giving you feedback. Let's get back to work. Okay. Let's talk about insulin and its regulation. A couple of simple tables, but these tables are critical for the exam. Take notes. I don't ask you to memorise too many tables, but this one's really important. Insulin stimulates what? Glucose uptake. Why? More glucose transporters. It does it in the liver, does it in muscle, does it in fat cells. Glycogen synthesis occurs where? In liver and muscle. We've covered that already, but that's what insulin stimulates. Increases our glycolysis, particularly in liver and muscle. Why? Because you're going to turn that glucose into fat more than likely. You're going to turn it into other things as well. What are you doing? You're making fat. I've teased you all semester. Which is the target enzyme? Well, glucose transporters, bless you. The glucokinase, keeping that going, increasing expression of that molecule, that is gene, write down gene expression. Glycogen synthase, what are you doing in liver? Storing lots of glycogen because insulin told you to. The glycogen phosphorylase says don't break it down, so insulin said that enzyme stopped working. What have you done to PFK1? Increased by altering our friend PFK2. We're hopefully getting some machinations back from that. These other guys we increase as well. Who's the counter-regulatory hormone? Glucagon. Insulin, blood glucose. What happens? Glucose declines. What happens? Glucagon starts coming up. Right on this page, there's no zero glucagon and no zero insulin. There's always a little bit in your bloodstream of both of those. It's like the signal from the kitchen. It's like your phone. There's always a signal saying which one. It's just how loud they get. What does glucagon do? Essentially the opposite. Just note that, essentially the opposite. Everybody, insulin stimulates, glucagon tends to inhibit, puts glucose transporters back in, alters the pathway of our liver. What are we doing? We're regulating metabolism from a hormonal level. Glucagon has its receptor too. Don't have time to go into it in this unit. We done with this one? Yep. Spend more time on it, get to know it. The problem for you, which is going to come back and bite you for Module 5, is measuring hormonal changes. And these hormonal changes are often circadian. Means they change over the day. I want you to look down the bottom of this graph, or both of them. 8:30 a.m. Half the day's gone by then, but most of you, that's about when you're crawling out of bed, isn't it? Not all. There you've got 12 noon, 5 p.m, 8:30 p.m, 12 a.m. What you're seeing is a circadian pattern of what? Blood glucose. What do you do after you get up? After you go to the bathroom, maybe have a shower, go for a run, whatever, you often have breakfast. What happens? Your blood glucose goes up. That would be your general routine. Some people skip breakfast. You might not see a glucose spike. You're seeing the amount of glucose in picomoles per litre. Take note of the units. Down here, what do we see? Insulin in picomoles per litre. Take note of the units. NADH, we're asking for picomoles. Down here we see insulin. What do you notice about insulin? It kind of spikes the same time, doesn't it? But just write down just after the glucose spike. As glucose increases, insulin starts to chase it up. But it's a little bit lagging because what happens? If I go back to the slide with our beta cell, you have to go through the glucose transporter, through glycolysis, then the citric acid cycle, the ETC, change the potassium channel, alter charge, increase calcium, and then you release insulin. That takes time. This is a circadian rhythm. Breakfast, lunch, dinner, maybe an afternoon snack if you live on college. When I lived on college, we had a 10 o'clock snack of cake and tea and stuff like that. Claimed we were going to go back and work, but never did. Now, this is also overlaid by the diurnal rhythm. Are we done with this slide? Okay. And that diurnal rhythm is bringing in other signals, other hormones. Here's one. Insulin we've just covered. What does it do? It promotes fuel storage. It promotes growth. Write down, it's a growth stimulator. It's giving cells the energy they need to grow. More stem cells, for example. Glucagon, what does it do? It mobilises glucose from stored tissue. We've seen that before, particularly the liver, to punch out more glucose. As we go through the next few weeks, we're going to see that evolve when we go through more and more fasting, for example. Underneath epinephrine, you're going to write down adrenaline. What does adrenaline do? It mobilises fuel very quickly, right down acute, in the matter of seconds and minutes. So every time you have a stress, a perceived stress of a metabolism exam, joking or otherwise, you will have a adrenaline release. Just note, please note, that's a brain perception, a neuronal signal to your adrenal, and a release of adrenaline, unlike some other hormones we're going to talk about. Cortisol, you're going to write down steroid. Does cortisol have a cell surface receptor? No, adrenaline does. So it's got to get into the cell cytoplasm. It's going to be carried around by something like a protein called transcortin. It's going to get in, it's going to alter long term. You're going to write under that chronic. It's a chronic stress hormone. It works over days, weeks, months, years. Can you have the two, an adrenaline spike and a cortisol spike? Yes, you do. And they can interact with each other, altering. Either you become more anorexic or more obese, depending on the response your cells take, which is what we're going to talk about over the next few weeks. We done with this one? No? It's going to be tough today. Having them delay our start and then lots of feedback.
Maybe the semester exam is out of 12 and up.
Mid-semester exam, let's see if it was out of 10. Look at the views notices. Are we ready now? Yes, mate. Just stretching, okay. Glad you're relaxed. Okay, everybody else all right? Okay. All right, why don't you look at the schema on the left. Don't write, look at the schema on the left. What do you see? You see your brain at the top, write down brain, CNS, central nervous system. What's input to your brain? Fear of me and metabolism. Fear and loathing. It's a film, isn't it? Pain. I'll come around and squeeze you so much it's painful. That's a neuronal signal as well. They are inputs. So is hemorrhage. What does that mean? Blood loss. What about that? Hypoglycemia. What does that mean? Low blood sugar. Brain says there's a problemo. You run down here. What does the green triangle mean? It means any of these signals to your brain, you're sending a stress response down to the hypothalamus, which is at the base of your brain. Kind of hangs down like a big oversized balloon. Now, I need you to jot down, the brain is neuronal inputs from your senses, from your perceptions. That's converted to a neuronal signal, that is a sodium action potential to your hypothalamus. You're going to write down the hypothalamus turns a signal from neurons into a hormonal signal. The hypothalamus produces these hormones. Here's one, corticotropin, or PHIN, you can spell it two ways, releasing hormone. So your hypothalamus at the base of your brain gets neuronal signals but releases hormones. Write down releasing hormones. That's what it releases. Those hormones are released into the blood vasculature to go straight to your pituitary, right in behind your nose. If you have a pituitary tumour, they actually go in through your nose, break a few bones, pull out the tumour. But your pituitary hangs down like a little punching bag. I want you to jot down, it has an anterior part and a bottom part, anterior and posterior. The anterior produces lots of hormones, so does the posterior. We're going to talk about those over the next few weeks. If you have a stress response of any of these, your hypothalamus gets whacked. It says to your hypothalamus, release CRH. Hypothalamus releases that hormone, which sticks to its receptor, write down receptor, on the anterior pituitary. That receptor stimulation signals the anterior pituitary to release hormone, in this case, ACTH. You write down 1 to 39 amino acids for a start. That comes from a monster polypeptide. I'm going to show you that next week. A big, long one. It's fragmented, stuck into vesicles like insulin. You're going to release ACTH. What does that do? Get into your bloodstream. You write down bloodstream here. When it goes to your bloodstream, it then circulates to your adrenal. Where's your adrenal? just sitting on your kidneys in that fat pad. The medulla produces adrenaline. The cortex, the outside, produces a number of other steroids, but some of them produce cortisol. That ACTH zooms down there. It has a fairly short half-life, fairly unstable molecule, about 5 or 10 minutes at the most, hits the receptor on the adrenal and tells the adrenal cortex, write it down, to release cortisol stored in vesicles. Dumped out cortisol, what have you done? That is a stress response signalling a hormonal, write down, cascade. That cascade has stimulated cortisol, which alters functioning of liver, other cells. And what does cortisol act as? Transcriptional regulator. Write down, alters gene expression. Now we have all these effects, so we're going to alter gene expression now. Do you see adrenaline on this? No. That can happen as well. I want you to jot it down. Any of these stresses, like pain or fear, that stimulates adrenaline as well. So you have an acute and chronic stimulation of stress hormones, which are going to alter your metabolism. And why is cortisol so important? Because in Module 5, you're going to measure cortisol of a patient that might be going through a stress event, either perceived, physical, or biochemical. You have to diagnose that patient. You have to measure the cortisol and their protein concentration. Cortisol does a lot of things, but you see in there, in exclamation marks, causes adrenal fatigue. If I keep getting stresses, what does cortisol do? Can't make enough anymore, can't produce enough anymore. I just don't do it. I actually get tired of making that hormone. That's down regulation. Write it down again. We're done with this one. Is that a yes? Yes. Thank you. Let's have a look at your adrenal. Now, you're going to write on this diagram that the colours are incorrect. You're going to write down the middle bit, the medulla, is actually white. You're going to write down the periphery, the cortex, is actually brown, because the white stuff is derived from neuronal tissue, which is white. The brown stuff has lots of chromophores making cortisol. What happens in this particular case? Well, let's have a stress response, shall we? See what I mean? Brown on the outside, white in the middle. What have you just caused in this effect? What have I released from the adrenal medulla? Adrenaline, write it down. Adrenaline's being released from the adrenal medulla. So my brain perceives something, or something happened physically. I've got a neuronal signal to my adrenal medulla. I've stimulated release of adrenaline. So now I have an acute response. I'm altering cell metabolism. Heart's racing, blood's racing, lots of glucose racing. Let's go and see what happens in my cell. What am I seeing here? Here's the cytoplasm of the cell. There's the nucleus. Here's the adrenaline receptor. You're going to write down beta 2 AR, the beta 2 adrenergic receptor. There's a number of them. There's whole families of these characters. Makes life very challenging. Your drug might affect this particular receptor. How many receptors? How much binding? Well, let's see what happens when adrenaline sticks to its receptor. Bless you. There comes adrenaline. Now remember, there could be multiple receptors, lots of signalling, lots of regulation. What does my adrenaline do? I want you to just jot down, that goes in and out of the membrane 7 times. Not just a big lump. This is a G protein coupled receptor. There goes adrenaline. Which cells are we on? Liver. I just altered my G protein, like you saw in that beautiful video. GDP, GTP, we dissociate GS-alpha. That runs along to who? Someone like adenyl cyclase, for example. Here he comes, AC. Alters him. What does he often do? Makes cyclic AMP. What do I do in this cell? Well, I get lots of calcium and my cell gets busy. I've just had a stress response. What did I do to that particular liver cell? What should I do to that liver cell if I'm adrenaline? Should I get it to make fat or to punch out glucose through gluconeogenesis? The latter. Jot it down. But at the same time I'm doing that, I'm going to actually get into the nucleus. Let's quickly look at this. Lots of cyclic AMP binds to cyclic AMP proteins in the cytoplasm of your liver cell. It changes conformation of regulated proteins. That's all these C proteins, R proteins. Some of those are going to get inside the nucleus through the nuclear pores. Excuse me. Binding to a cyclic AMP response binding protein. What do you know? Phosphorylation again. This protein is now active. What does it do? You're going to jot down that's a piece of DNA. That DNA has a transcriptional regulatory region called the cyclic AMP response binding element. Once those two get together, there's a marriage made in heaven. They start producing lots of mRNA. Sometimes it's a bit slow, it's 9-month gestation before parturition. I'm making lots of mRNA. If we keep acutely stressing you, like a lecture every week in metabolism, what happens? Heart rate up, whatever. Okay, I get more glucose out. But acute stimulation more constantly causes this effect where we get this signal down to the nucleus. We start making messenger RNA. Because you're acutely stressed, making lots of glucose through gluconeogenesis, your factories are being told, put more machinery in there for gluconeogenesis. Does that make sense? So you're now priming your system all the time. You're pushing it to gluconeogenesis because you're a stress head. If you relaxed and saw metabolism for what it was, which is pretty straightforward and easy, you wouldn't be so stressed, would you? But what you're doing now is inducing gluconeogenic proteins and enzymes to make glucose anytime you have that acute stress. Does that make sense? Okay. That's an acute stress. What does cortisol do? Well, cortisol's a royal devil. It's biphasic. Write that down. It can do good and bad. A little bit of good and often a lot of bad, which we're going to keep talking about during the semester. These are important to us because we have an acute response overlaying a chronic response as well. Kind of comfortable with that? Is that a yes? Can I move to the next one? Okay. Now I said you have a diurnal rhythm. I'm going to come back to why in greater detail. But it's because of how much light and dark you get in the real world. Through winter, you have lots of darkness, don't you? Look down here on this scale. This is the amount of light over a 24-hour period. Here's the amount of dark. Cortisol is linked to not just your stress response, but to the light-dark cycle. If you look here, you can see cortisol drops in the early afternoon, in the evening and nighttime, and just around the time that you start waking up, it peaks, then it drops off again. You're going to jot down, if I get stressed, watch my laser during the day, that might shoot up. But that's your normal circadian, what we call diurnal rhythm. Evening drop off, late night, mid-morning, get ready, that's important for the next few weeks, and then the day goes on, overlaid with stress responses. Down below you see adrenaline, it's kind of similar, isn't it? And a stress response, adrenaline goes up, comes back down again. This is your normal circadian profile. When you take a blood sample, it's critical during the day to overlay the circadian or diurnal rhythm. Does that make sense? Do proteins have diurnal rhythm? Does your functional glucose transporters have diurnal? Yes, they do. We're going to talk about it over the next few weeks. That's critical for us. I'm going to keep coming back to this circadian pattern because you get ready to wake up. You get your metabolism ready to do work, even though you don't know it. Can I get to the next one? Okay. I need to talk about this. I'm afraid we're going to go a bit over time today because of our predecessors. Module 5. These web links will be up on Views, 1 o'clock on Thursday. Don't have to watch them now. Next week, if you're not on this campus, sorry, if you are on this campus in Hawkesbury and you're Wednesday at Parramatta, next week you have off, don't you? Poor old Parramatta Tuesday had a kind of timetable issue. They've got 1 week behind. But next week you have off. Next week you should watch these videos whenever it suits you. It's up to you. They'll be up Thursday at 1 o'clock, stay up till the rest of the semester. It's important to watch them because one tells you how to do a cortisol competitive ELISA. The other one tells you how to take the data and process it to get a value, a concentration of cortisol. We're not going to do that in the lab. You need to do it before you get to the lab. Because in the lab, we're going to test whether you can watch this particular video, follow the instructions. And the instructions are you're going to get a saliva sample. You're going to measure the cortisol in that sample. You'll probably do it in pairs, simply because, as I said last time, 96 well plate, break 8 wells in half, 4 wells, 2 wells, 2 wells. Once they're in your hot little hands, NMP, your problemo. Demonstrator will do the standard curve, tell you when to do things, that's up to you. Everybody will do a protein assay. They're going to use the sigma protein assay. Sigma protein assay to measure the amount of protein in that saliva as well. You will need to demonstrate to the supervisor and demonstrate that you did your own protein standard, not someone else's. You must do your own. Is that kind of clear? Show your own. Your patient's protein and your patient's cortisol are worth 1% each. You're going to calculate that and you're going to go to views. There's no lab bookmarks now, just the views question.